Thanks for all the speakers so far for keeping to time. Something really resonated with me, that Suzanne mentioned, and Sarah alluded to it, that you can go from 4.5 million compounds down to 6, 7, 8, 9, which is where we are today, all having high potential. A big barrier, of course, is the physicochemical properties of the chemistries. If you can't put it on a net and keep it there, and make it bioavailable and make it wash resistance, that's an additional challenge that frequently doesn't occur in agriculture. So this is why it takes 4 million compounds to get to just a small handful. So I'm very pleased to introduce Amir Galili of Weston. Excited to have been working with Amir and his team for the last couple of years on what we think is a high potential project. It's not something that's going to come to market immediately, but it's within the foreseeable future, we believe. Amir founded Weston Company to develop and produce effective control tools for vector and nuisance mosquitoes. Weston is the developer and IP holder of several patents on ATSB technology. Prior to Weston, Amir founded and managed several communications and high-tech companies for more than 15 years. Amir holds a bachelor's and an MBA degree from Tel Aviv University, and we're happy he's joining us today. Thank you.
Thank you. Hello, everybody. I hope it's okay. This is the right side.
How about that?
Thank you. I would like to focus on the difficulties of development of a new tool. Absolutely new tool that is not following other type of tools. So this will be the main subject that I'm going to speak about. I know that the efficacy and many questions about non-targets and others will be very interesting, so I will briefly touch them, but most of the talk will be about the challenges of new tools. Most of the people here heard it probably several times about the sugar feeding method that we are working on, and the fact that mosquitoes need sugar, and in any case, even if they don't if they can survive without sugar, if we are suggesting them sugar, they will take it usually happily. And it's also for male and female. And based on this fact, we developed the product, the attack and kill method. This is enabled to decimate the mosquitoes. I believe that this slide is one of the key points of the development. This is where we have to see if it's able to have a control method based on the need of mosquitoes, the sugar need of mosquitoes, which was known quite a long time, but that was the first time that we tried to prove that it's possible to base a method on this one. So in early trials by Dr. Muller and Professor Schlein from the Hebrew University, they sprayed different plants with colored sugar solution to identify the important sugar feeding. And they found out that high percentage of the mosquitoes are colored. Right after that, the attractive plants were sprayed with toxic sugar, which significantly decreased the number of the mosquitoes. Those two facts basically shows that it is possible to make something out of the sugar feeding. But there was a very long way in order to have a viable product following this one. I think Sarah mentioned some of the idea, what do you need for a new product? Here we had few more challenges apart from all the toxicity and the formulation. We have to show the appropriate attraction distance to utilize with variety of active ingredients, to be able to manufacture it, to have the stability of the shelf life and the active life in the field. We need the right application, which is a big one. We'll show it later. As I mentioned, a minimal impact of non-target because we are going to put sugar in the field, and to make it economically produce it, to make it enough time working over 6 months, and to put patent and to be able to register it. I'm sure there are other items, but those were the most important ones. So after we identified that it's possible to make a product out of it, we try to look for the most different in our product, and this is the attraction. So we have to discover the right bait. Initially, Dr. Muller and others made a homemade food-based bait that was okay, but it's impossible to work with it in the field and to make it a commercially available product. So we have to find something that it will be able to make it commercialized. We brought into our lab hundreds of possible attraction. Some of them work somehow, but they all wasn't in the right price or in the right quantity or couldn't hold for enough time. We utilize GC, electronic antenna graphics detection. We used olfactometers. We use room attraction in order to identify the bait. And finally, after, I would say, it take us at least 4 years to identify and to look for the right bait, we currently have a product that it's a combination of fruit extract and with some other volatiles added to. I would say that this was one of the main issues that we worked on. I don't say it was a challenge because it's a very straightforward, but it's a lot of work in order to identify. Then we have to identify the right active ingredient, the ingestion toxicant. We had what we need. We need to have the efficacy, the stability, to have the regulatory status, and of course the right supporting company. We find few active ingredients that are, I would say, natural, that in the US they are 25B, but they will not be, I'm afraid, strong enough for the US. So currently in the US, the active ingredient we are using is dinotefuran by Mitsui. So at this point, we established that the sugar bait is valid solution. This was the most important part. We identified, I would say, the 4 parts of the bait: the attractant, the active ingredient, the sugar, and what we call the bait stabilization, that it's combination of everything that is not attractant, not active ingredient, and not sugar, like slow release, structured definer, and preservatives, and some other. This was the sample. And we managed to demonstrate some of the ATSB advantages. For example, the main one, I would say that it's the chance to develop resistance is much lower because here we are really speaking about new mode of action, because it's as we said, it's not a contact poison. This is number one. And few more advantages that you can see here. The next major challenge was the type of application. So we started with spray vegetation, which is very simple, and it's low in the production cost. I'd say very, very straightforward, and also a very high advantage is that the surface of the bait is very large when we are sprayed on the vegetation. However, vegetation is seasonal and it's not always available. This is one thing. The non-target is very much exposed when we are putting it on the vegetation. And We cannot use it for indoor and also very close to the house. But I would say that for the Africa environment, probably the major one is the lack of vegetation in the area, in large quantity within the villages. So we will need a different one. We will need a bait station, a type of feeding device. Again, here we had tens of type of bait station that we checked and try to activate. Here you can see some of them, and here few more that were a little bit more advanced one. The challenge is how to hold the bait in the station, because I would say it is impossible to put the bait on the station and to hold it outside in the rain if we want it to have it vertically. How to ensure a minimum product service life for 6 months, how to manufacture in the right price, how to protect the bait from the environment, and how to make what is the best design for a simple bait station, basically two dimension. The two dimension is critical. Maybe it's not so much understood, but the two dimension, it's very important if you would like to put a large number of bait stations within the village. Just to put it, if it's not two dimension, we have to put it on a stick, which is another issue, and the whole village will have bait stations all around it. The game changer was the development of the membrane. We got into idea that the only way to make it two-dimension and also to answer all the rest of the questions that we have here was to develop a membrane. And that's what we did. Again, a very long process of developing a new membrane. We initially were looking for membrane existing on all type of filter papers, all type of what is using in labs for blood feeding. None of them was good enough. Sometimes it was the temperature. For example, we start with silicone, but silicone is not stable for in 40 degrees that it might be, 45 degrees. Also the cost is another issue the problem, we wanted to stay with the bait inside and not getting dry. A lot of the membrane become dry after a while. So we had to develop the membrane by ourselves, make it, I would say we make it from SEBS, that is the main ingredient within the membrane, but we have to develop also the process. So we have two-dimensional bait stations that can be hung on the walls for indoor and outdoor. We have thousands of micro holes within the membrane, approximately 10 micro each one of the holes. It's black because we want to protect it from the UV, and it's quite an advantage for the mosquitoes. The bait is protected from the rain, dust, and the animals. And also, the bait is inaccessible for children and pets. That was important if we were going to put it within the house. And we extend the life of the bait station for over 6 months. We believe we also managed to reduce the cost, because whenever we put something with a rigid plastic into it, the cost was 3, 4, 5 times more than what we reached here. And we believe we will be able to manufacture it, the cost far behind $1 with a large number, which is very important in this case. So, the current status, okay? Following 9 years, a lot of money, and plenty of dead ends. By the way, what I'm describing here is without focusing, of course, on all the dead ends and the mistakes that we made. Quite a lot. We have a spray application that is commercialized 4 years ago for the retail and the PCO within the US. The spray application is only with 25B, only natural active ingredient, not dinotefuran. And we have the bait station application that is currently on testing. Currently, we are conducting the outdoor bait station experiments in Mali. Thank you, Nick, again. Sponsored by IVCC, aiming to validate the benefit of the ATSB method for vector control. Currently, we are getting into more epi trial and more entomological trial, and we hope to complete it toward the end of 2020, but it will be in the market 1 or 2 years later, of course, if everything will be okay. And also, we have the initial results of indoor experiments is very encouraging. That this is also something that we are doing. Both of them we are doing in Mali with a lot of help of Bamako University. Now a little bit about some data and some issues, and as I call it, frequently asked questions. This is about the efficacy. We have sweet level. This is just a sample of the efficacy. We have it's quite, I would say it's tens, if not hundreds of type of experiments. The basic one, of course, are the cages to see that they are eating, to see that the mortality of the mosquitoes. We tested the PPN, Albopictus, and the most important one, Anopheles gambia, that we cannot test it in our lab in Israel, and those have been done in Mali with Anopheles gambia. And the efficacy is good, and usually it's after either 24 or 48 hours. Again, in this case, sometimes it takes longer for, to reach the above 95% mortality. That this is our aim. In the room that we test, again, this is from Culex PPN. We just put it in the room. We tested it. It was very important for us to make sure that the bait station will last effectively for over 6 months. So we put it outside in the sun, and every 2 weeks we put it back, and we showed there was some decrease after 6 months, but it was no more than 10 or 15% reduction of the efficacy. And we had quite a lot of field tests, and I think that the most important one is the one that's going on currently in Mali with Anopheles gambia. And it shows, I will show it a little bit later, just in 2 more. This is just type of efficacy, a room efficacy that we made with the bait stations, in this case, with Anopheles PPN and with Culex PPN and with Albopictus, as you can see. And after 48 hours, the results are extremely good within the room, but the outdoor is totally different issue within the outdoor. But you can see that again, over 95% after 48 hours. This is, and I would like to emphasize, this is interim solution only from our current entomological experiment in Mali. There is a big issue about, I will not get too deep into it. This is under VCAG, and we presented it also to VCAG. The full experiments and the full report will be ready within, I would say, 3 to 4 months. The important part in this case is to see the reduction. That's what we believe, to see the reduction of the old female mosquitoes, those that can transmit malaria. So you can see that we have here, I would say in all of them, either EIR or the spore site or with the old mosquitoes, managed to reach around 90% mortality. Now, we don't have much time, so I'll just go very quick. The non-target and pollinators, we know that pollinators are not attracted to the bait fully, and the membrane protect from the pollinator. We should go through all process of regulation, and we start with the EPA in one hand and with the WUPAS, later on PQ on the other. I will not go over the production technology, but we started from the beginning. We developed the full production of the current process. And of course, as Nick mentioned, we have few patents on the bait station and various on the technology, on the bait, and on the method of killing mosquitoes with sugar. And the last slide, we developed a patent-protected bait formulation and membrane and the bait stations which effectively control mosquitoes. What is our expectations? Our expectation, I put it very bold, to improve what we have up till now. We are sure it's possible to get even more attractive bait than we have, and we have to learn still how exactly to operate the bait station. So improvement should be done. We are the first one to do it. We did it by ourselves. There is a huge place of improvement, and we're sure about it. We expect that we will be able to make outdoor malaria vector control, indoor malaria vector control, Aedes control for indoor and outdoor in other places, sandflies control. Actually, the initial experiment was started with sandflies. And I'm to work with the ATSB to manage resistance, but I will not go over it too deep at this point. And we expect ATSB to become a significant component of the vector control strategies, as it can deploy both indoor and outdoor, and it's less likely to promote resistance. And I would like to thank you that here, IVCC, and of course, specifically Bamako University and the team that worked in the last 2 and more years in Mali and did a great job. Thank you. This is yours.
Huge amount of potential here, and we're very excited to be a part of it. While I'm bringing everybody up here for a panel discussion, there's a bit of a consistent theme here coming through, and that is that product development requires patient optimism as well as impatient optimism, talking about Bill Gates. If you look at the timelines, 19 years to develop pyrethroid nets, 15 years for a novel AI, 9 years with 4 years to go for an ATSB. So it takes organizations that are, and I must say, like our funders, like the Gates Foundation, who are prepared to stay the course on something that is long and painful and sometimes outlives people in those organizations. So they need the staying power to go forward. So we're open now to questions. We have about 20 minutes. There's a microphone here, one on the other side. Maybe if you'd like, somebody would like to go first, or I'll claim chairman's privilege perhaps and start things off. I'll ask Amir a question since he was last to go. Amir, we're finding out that efficacy is better with older mosquitoes. Are you prepared to comment on that at the moment?
Yeah. Basically, the mosquitoes, when they are getting into the houses and outside of the houses, crossing the villages. And when they're crossing the villages, and as the bait stations are hanged outside for outdoor activities, they are hanged on the wall, their chances after they get in and out a few times is much larger to be caught by the bait stations. And the only thing we need that one time when they pass next to the bait stations, they will test, feed just once, and that will be enough. That's why there is a difference between the general population reduction and the old female mosquito reduction, and that was, and this is important.
Thank you. So we'll take the first question. There's a microphone up here, John.
Yeah, sorry, Amir, another question for you. Have you done any social study on how long these would survive hung on houses over 6 months? I mean, without intervention, if they were put there, are they removed, taken away because they're a novelty or of interest? And what about African villages are very dusty. I just wonder if with a sugar attractant they become very dusty and ineffective.
Thanks to IVCC, and actually not only them, there is a huge work done about the social science. This is important almost as, sometimes I think, almost as killing the mosquitoes is to make sure that the acceptance. So there is a lot of activities in this field. By and large, they are very well accepted. However, we learned that we have to improve the communication with the villages. In at least 2 cases, there was some issue within the villages that some didn't want to put it, but it was identified. It was just the issue of communication. 6 months, if it's, again, we learn how to hang it better. Initially, the hang wasn't good enough. It was good, but sometimes it failed, and then it was improved. It's possible to put it, and it will hold for 6 months. It's possible. And regarding the dust is inaccessible to the bait. I would say that more important is the rain. Usually we are speaking about the rainy season, at least, again, we are speaking in Mali. So it might be other finding in other places. More important is how to make sure that the rain will not harm the bait station. That's what. The dust is inaccessible, and then the rain is coming and it's totally washed out, if there is a dust. Okay.
Thank you.
Yeah, thank you. My name is Godwin. Did you consider adding synthetic odor to improve the efficiency of the sugar beet tetra?
We will be very happy to find any odor to add it. And if there's anybody here has some idea or more than idea about attraction, we will be more than happy. As I said, we're sure it's possible to improve, especially the attraction.
Maybe I'll just add to that. I mean, and it goes back to my comment about how long it takes. Product development really does take. At some point, you can go on tweaking these things forever, but also at some point, you have to pull the trigger and try and get the first generation to market. And I think that's really where we are now. But in parallel, there's a real opportunity to take what already is a very interesting looking and potentially disruptive technology and make it better. And one of the benefits of having a conference and being able to present at this is something like this is that we will find folks in the audience who have ideas or would like to work or support us in thinking about what Generation 2 might look like. Anyway, we have time to take another question.
A question for Sumitomo and BASF about resistance management and responsibilities for this. So you talked about the importance of producing these new chemistries, which are an excellent step forward for us being able to maintain control against resistant mosquito populations. But could you say something about where you see industry's role in ensuring the appropriate stewardship of these chemistries? And one thing is to get the chemistry, and the other thing is to use it to ensure that we do manage resistance.
Okay, so this is a very extremely good question. How does industry try and sort of regulate its own chemistry? I think my own view and my own personal view, industry, as you know, Henry, is very involved in Iraq, and we totally endorse the concept and idea of rotational strategies because it's to our own interest to make sure that our products the life of the products are maximised. But it's not up to us to decide who and when our products should be sold. Our interest is to sell product. And while we endorse rotational strategies, it's really up to countries, and it's up to WHO, and it's up to other organizations to implement those rotational strategies. So, for example, we work with PMI, and PMI should be saying, right, well, we'll buy your product this year, and we'll buy a different product another year. Our interest has got to be to promote product. Obviously, with all good stewardship and making sure the label recommendation is endorsed and all of that, that's a very important part. But for as soon as we start to say, oh, well, we'll sell our product in this country this year, but we won't sell it next year, I think that's, what if a country says, but we want your product? We actually need your product in that country for a whole range of reasons, and we'll perhaps rotate on a 2-year basis, or we'll do something different. As soon as industry comes up and says, well, actually, okay, we won't sell this product in this country because we sold it there last year, what do we then do if a competitor comes in with the same mode of action chemistry and says, oh, great, fantastic, the Sumitomo has walked away, we'll take the business. And the country says, oh, great, we'll buy your product. So it's almost a conflict of interest although our bigger interest is to endorse rotation and support it, and we all talk about it, and we're all part of Iraq and all totally agree with it, we also are pushing and working with other stakeholders to say, guys, WHO, countries, PMI, you have got to implement those strategies, not us. I think that would be that's my comment. I don't know if you want to add to that.
I mean, we also encourage rotation because it makes a product live longer. Yeah, it's also about keeping it in the market and active for a long time for doing business. I mean, we are as industry to set up to sell things. We produce it. We take the responsibility to make sure that it's safe to use. We give recommendations how to use it. As he said, we give recommendations what we would like to see in the field. But it's also in our interest that we accept that in one country we only sell it one or the other year. So you might sell it in year 1 and then you don't sell it and sell it in year 3 or 4. We encourage rotation, although we lose business in certain years, but in the total you make your product last longer. So it's in both interests. Yeah, it's you keep a weapon sharp, and on the same time you also keep your business going on. So it's in our interest too to do rotations.
I mean, if we had enough chemistries. Yeah. For example, if one company had a huge range of chemistries, which we don't, we've seen we have a paucity of them, then possibly a company could say, well, okay, you'll do a deal, we'll sell you this product this year, this product, a different product the next year, but we don't have that luxury. So I think it's really incumbent on countries and programs to really implement those rotational strategies, and we will totally support that.
John, it might be worth just saying very quickly what IRAC is for those who are not familiar with it, and that agriculture has been dealing with this for about 50 years.
Yeah. Insecticide Resistance Action Committee is called IRAC, and there's a number of different ones. There's a fungicide one called FRAC, there's a herbicide one, and Sumitomo and BASF and Bayer and all companies are part of CropLife International, which host the IRAC activity, and there are a number of bits of information, literature, and there's a mode of action chart that is produced. So we are actually in the process of making sure that mode of action chemistry is actually indicated on labels in the ag business, but that will flow through into public health as well. So people buying products can see what the mode of action chemistry is. But there's lots of guidelines about rotational strategies. And IRAC have even funded many years ago now in Mexico a large study looking at the benefits of rotation and mosaics and generating data, evidence-based information.
Thank you. I think we've got time for one, possibly two more questions.
Hi, maybe I can add something to the discussion of keeping active ingredients into the market and preventing resistance. I think here is one very important tool is that we have a susceptibility monitoring, and new active ingredients can have different characteristics and maybe not behave like a pyrethroid in common susceptibility monitoring tools. And here I would like to promote a little bit the poster 187 downstairs, where we show how to make a CDC bottle assay with clothianidine, especially, because that is a bit different to the standard bottle assay we know. And I think this is another contribution to keep active ingredients or to maintain active ingredients in the market.
I think that, thank you very much for making that point. And that's just to add to that. Of course, PMI, as one of the, an ABT as an implement of the PMI, do in every country do susceptibility assays before deciding on which chemistry to use. And Sumitomo and of course Bayer are working on looking at clothianidine assays, bottle assays, and making sure that we have a diagnostic dose that PMI can work with, so that they ensure that if there is any sign of resistance to our chemistry in the country, they then use a different chemistry. That's the principle.
I think susceptibility monitoring is essential. On the other hand, what we're trying to do here is make sure we don't get to that point. Yeah. Once you're starting to see resistance kick in, you could just claim we failed. What we really need to do is be rotating these chemistries properly up front, and then we're not going to have that issue. I'm happy to take another question, and then we'll probably have to wrap this up.
Paul Sykes, thank you very much for the presentations. In the first presentation, cost and affordability issues was mentioned as one of the criteria for sort of assessing the potential impact, benefit of these products. I wonder if we could ask the three who did present very interesting new products to touch on the cost issue as far as you can make sense of that at this point, so we understand perhaps the implications of these products.
Cost of the product or cost of the development?
Well, I guess from a programmatic point of view, I had very dismaying experience of, like a lot of us, looking at the implication of losing pyrethroids in IRS and how you then might have to scale back the coverage of IRS programs. From a programmatic point of view, trying to reach large populations, what would these products mean in terms of affordability?
Can I comment on the IRS one? Just on the IRS thing, I mean, we're working very closely with the engineers program funded under IBCC, and they're looking at trying to introduce product into market in an affordable manner. Did one of you guys want to say something about that? I think, David, I don't want to put you on the spot, but I think it would be appropriate for an engineers person to make that comment, because it's very valid. Yeah, I mean, the question, really, the concern is that new chemistry and Sumitomo's Sumishield is not as cheap as a pyrethroid, but nor is Actelic, and nor is Ficam, and nor is Bendarkite. So those chemistries are more expensive, and there is a risk of reduced coverage, but there is Sumitomo are working with engineers to try and introduce these products in a manner which makes them more affordable and cost effective through a co-payment scheme, and then we're endeavouring over the next few years to try and really drive down our cost to make these products at least not dissimilar in cost to, say, the organophosphates that are currently used in the marketplace. But it is a concern. I mean, it's all very well to say we need new chemistry, but if it comes at a huge cost and they're not affordable, and that, I guess, Sarah, you were talking about as well in that gleaning process of all those different 4 million molecules or whatever it was, and you get down to 27 chemistries. Some of those might have been thrown out due to just absolutely immensely high cost of production, and if they're really too expensive. And that is one of the criteria.
Yes, absolutely.
I think the idea that we're ever going to get back to chemistry, certainly in the short term, that's the same price as a pyrethroid, which is very low dose rates and cheap as chips, is unrealistic. And so somehow we're going to have to manage that. But I think technology can also manage that. So let me just speculate. If you can get a novel chemistry that would give you one year performance in an IRS, possibly 2-year performance in an IRS, that changes the entire economics of IRS. And those are the sorts of things we're looking to do today. Hence my comment about technology is not just about the active ingredient, it's also about the formulation chemistry and the delivery of it. And we're spending a lot of time thinking about that. So think something else here that I'm again speculating. If you could switch IRS for something akin to an ATSB, if you could use that indoors and outdoors, it's potentially even a consumer applied product, which might dramatically change the financial dynamics of the marketplace. But we're very, very conscious. So chemistry, for example, to us that is going to cost more than $150 per kilo, and sometimes it's extremely hard to know what these compounds are going to cost until you scale up, until you find a new method. Mostly we wouldn't even go there, because now you're talking about products that are in excess of the cost of Actelic. So cost is very much a part of the decision making of which chemistries to take forward. But it's always going to be a challenge, and I think we're going to have to be as creative in how we go to market. I mean, there are also why not potentially own a chemistry, make it and sell it at cost? Why not through some of the funders? There's another way to do it. Might undermine what manufacturing is doing, but I think there are creative ways to think about how we fund this, how we manage the funding gap going forward. Yeah.
And also for the LLINs, now after the learning with the IRS, with the lower coverage because of higher costs, they have set up a catalytic funding from the Global Fund. So they want to prepare for nets with other chemistry, which are more expensive, to come into the market and not be reduced in numbers. So there are plans now and money available to do better than it has been in the beginning with IRS. So there's a learning.
All of us are here this week. I know we have to wrap up now. It's dead on. I've got a red light flashing. So, but we're all here and happy to answer questions outside the meeting. But thank you all so very much for your patience and participating.